We'll wait a few more minutes to get started. There are quite a few people planning on attending. Well, thanks everybody for joining us this evening. This will prove to be quite an interesting program, I believe. Bob did this for us, what, about 6 years ago, Bob?
Yeah, something like that in Brantford.
In Brantford, and it was amazing. Next time I went out on a woods walk, there were all these things that just popped into my vision. So I figured that there are a lot of people that haven't seen it, so let's give it a go.
Okay.
So welcome, Bob, and thanks for joining us.
Glad to be here in the virtual sense. Okay, so I'm going to get started. I don't have a view of everybody who's here, so I'm assuming that you have most of the people you were expecting. Dennis? Oh, you're muted. Okay. Anyway.
We've got about half of them.
Half of them. Okay. Well, they can jump on whenever I guess they feel the inspiration to do so. All right. I'm going to be talking about fungi, and this is not an identification talk. I just want to emphasize that. I'm not here to tell you what things are named. I'm really talking about the ecological role that fungi play in the forest, because that's really where my interests lie. And by the way, now can you hear me now? I take it you can hear me. Nod your head.
Yes, we can hear you, Robert.
All right, thank you.
I muted you instead of myself. Sorry, Bob.
Wouldn't be the first time. All right, we're all learning here. All right, so anyway, what I was saying is that I'm going to be talking about the ecological role that fungi play in the forest. My interests in particular are in fungal pathogens. So some of what I'm going to be showing you are fungal pathogens. But what I was saying before I got muted was that this is not a talk where I'm going to be saying, here's the names of things you'll see in the forest. It's not my focus, or not where my interests primarily lie. There we go. So these are the types of things everybody thinks of when they see fungi. Some of them, we call these sort of the macro fungi of the forest. This is what you see when you go out there, but actually there are fungi out of sight that are playing a very, very important role in the forest that don't have this sort of gross morphology that invites casual onlookers. Not to say that they aren't beautiful, but there's far more. They are the tip of the iceberg as far as the roles that fungi play in the forest. They play roles as recyclers of nutrients, carbon, gases. They are pathogens. Again, that's where my interests principally lie. That's what I get paid to do, is work on fungal pathogens, mostly of the forest. Parasites, and as partners, as symbionts in relationship with plants. Now, I want to just take a few steps back, just to sort of put things in perspective. We're going to look here at the very, very large, sort of deep view of the phylogenetic tree of life. And what you're looking at here are all of the different bacteria and the archaea. These are all what people used to group together as just bacteria. And then there's the eukaryotes, and you'll see here that there's this little node here that has everything that we ever think about when we think about life on this planet. Our interest principally and our focus almost entirely are on animals, plants, and fungi. But there's way more going on in the world, and certainly making up a far greater amount of the total biomass in the world than just this tiny little branch here. But if we zoom in on that branch, because that's where our attention tends to be focused, now we're looking at that tree of eukaryotes. So we've knocked off all of the bacteria and the archaea. They're this little node here, even though they make up the vast majority of the biological diversity on this planet. And we have these large groups here. If we focus in here, we have plants, animals, and fungi, again, leaving behind a great deal of biological diversity that simply does not attract our attention. Here are the fungi on this branch, much more closely related to animals than either is to plants, even though historically plants and fungi were taught as a single course because they were not animals. That's back in the olden days. You were either an animal or not an animal, and if you were not an animal, you got taught as one large group of organisms. Now, if we go zoom in a little bit more on the fungi, we have, again, what attracts most people's attention when they are out in the forest are the basidiomycetes. The ascomycetes make up the majority of the plant pathogens. And then there's all the rest. Here's the animal branch here. We won't worry about that right now. Are these other groups of fungi that I won't even talk, well, I'll talk a little bit about the glomeromycota, but for the most part, we're gonna skip these and talk about the ascos and the basidios. So what distinguishes fungi from most of the other organisms? Well, first of all fungi are heterotrophic, so that is a characteristic that they have in common with animals, for example, and that is that they obtain their carbon source or sugars from other organisms. So what are some other heterotrophic species? Well, we can think of animals, of course, all of the different things that are animals, and many bacteria and archaea are also heterotrophs in the sense that they obtain their carbon sources from things other than themselves. And we contrast these with autotrophic organisms. These are organisms that synthesize their own sugars. And that, of course, makes up, I have a question mark here because when I give this lecture in person, I usually try to get response from the audience, but we're not going to do that now because it would slow us down here. But that's going to be plants and photosynthetic bacteria and also algae. So fungi secrete their enzyme, like animals, which for the most part, most animals, I should say, take food into their bodies and digest them internally. Fungi secrete enzymes outside of their bodies and absorb the digested nutrients. And again, I would ask the question if anybody knows what organisms also do this, but I'm going to tell you, and it is spiders. Spiders perform their heterotrophy in this manner as well. They kill their prey, they secrete enzymes into their prey, digest them into juices that they then just suck up out of back into their bodies. So in the fungi, there are many ways that carbon sources are exploited. They function as decomposers, or another term we use are saprobes, where they feed on dead organic material and wastes. They function as parasites and pathogens, absorbing nutrients from cells of living hosts, in many cases causing disease. And again, that's the area where most of my research focuses, or almost, I would say all of my research. And then also as symbionts, where they form mutually beneficial relationships with other organisms. And finally, there's a small group, I won't spend any time on that today, but fungi that actually do function as predators, where they can trap living organisms, kill them, and then eat them. But there are very rare examples of this in fungi, but they do exist. Now, of course, for my talk today, talking about fungi and their role in the forest, we're going to talk a lot about their role as decomposers, because this is a really important role that they play. And they do this both above ground through trunk and limb rots, forming cankers and canker rots as foliar pathogens, as vascular wilting organisms, and then also below ground as root and butt rotters. And today, I'm going to really be focusing on these three groups. I won't talk about vascular wilts. I won't talk about foliar pathogens. We'll focus on these three groups here that function as rotters. Now, trunk and limb rotters, most wood decay is actually caused by fungi, although it is often preceded by bacteria or archaea. But the principal or the principal function of breakdown of wood on the ground or even standing wood is by fungi. And in this sense, they play an essential part in forest ecology in nutrient cycling. Nutrient cycling would soon come to a complete halt were it not for all of the fungi that affect this process. And of course, part of that nutrient cycling involves the carbon cycle, which is to say gas exchange with the atmosphere. So we'll start off by talking about trunk and limb rots. Two major components of wood important to fungal rot, and this is just something to keep in mind. Wood is made up principally of cellulose and hemicellulose, and that on average, this varies to some degree, is about 75% of wood. And this is the part of wood that we think of as being white. So that's the cellulose and hemicellulose. And about 25% of wood, and again, there's some variation here, is lignin, and that is more or less brown. Structurally, you can think, if you think of concrete and think of the rebar that is in concrete that provides structural integrity to concrete, the cellulose and hemicellulose would be the rebar, and the lignin would be the cement or the concrete that surrounds the rebar. So think of it that way when we talk about breakdown of wood. And this is important when we discuss these three types of wood decays: white rot, brown rot, and a smaller group of fungi that affect a process called soft rot. And often you'll hear forest pathologists talk about white rotters and brown rotters, and not very often talk about soft rotters, and you'll see why in a moment. Okay, so white rotters. They're white rotters. These are all white rotters are in the group called Basidiomycetes, and these are fungi that preferentially metabolize lignin. So they will break down lignin and metabolize it and leave behind cellulose, the white part, which remains. Other fungi will come in then, or maybe some of these white rotters will start to break down cellulose, but there's often a progression of different species that are present during different stages of decay of wood. And again, this can be either wood that's on the ground, we call that coarse woody debris. A branch falls on the ground, a tree falls down, a tree dies and leaves behind roots. All of that breakdown of wood in the tree and the roots is affected by these various stages of rot. And the effect of white rot is because it leaves behind cellulose, the lignin is largely metabolized, it is fibrous. And we'll look at a picture of this in a moment. Brown rot, on the other hand, also caused by Basidiomycetes, involves the preferential breakdown of the celluloses, the cellulose and hemicellulose, leaving behind the lignin more or less intact, which is then broken down by other fungi that come in following the brown rotters. In this case, because the cellulose has been broken down, it's lacking that rebar, if you will. The wood shrinks and cracks and forms these cube-shaped chunks. And so for this reason, it is often called cubical rot. And then soft rot, which is not as commonly found in the forest anyway, is caused by ascomycetes, some species in the Ascomycetes. And this is similar to white rot, but it is much more important in what we call woods and service, which is lumber. This is not a process that we often see in the forest, so I won't talk about it today. White rot, we'll go back now, look at some pictures here, very fibrous. So you can see here, if you are in the woods and see a branch on the ground, you can watch it, you can't watch it, but if you go back month after month, year after year, you can see this process and you'll see this fibrous material remaining. Looking at it, you can hardly believe that it could have held itself together as a tree, you know, several tons of material defying gravity, but that's because the concrete part of this relationship, the rebar and concrete, is gone. The lignin has been metabolized, leaving behind only the fibers. And this is what is taking place in this cross-section here. This is what was heartwood after the process or during the process of white rot, leaving behind this white material, breaking down the lignin. Some species that are involved, and there's many, this white cheese polypore, Tyromyces. This is a fungus you'll often see on standing stumps, dryad saddle it's called. This is a white rotter. This willow bracket fungus, Phylinus igniarius, is also a white rotter. And there's no way you can tell by looking at the fungal fruiting bodies, which is just a tiny, small portion of the fungus. When you see the fruiting bodies, removing it does not remove the fungus that is causing the rot. That'd be like saying removing an apple is equivalent to removing the tree that it grows on. These are just fruiting structures. So the main business of this fungus is inside this tree doing its work. Turkey-tailed Trametes is a white rot fungus. Ganoderma is a white rot fungus. This bright Cinnabarinus, Pycnoporus cinnabarinus, is a white rotter. They're really common. You'll see them on down wood. Brown rot is the one that I called also cubical rot. This is where cellulose is metabolized first and broken down, leaving behind only lignin. So it is as if you magically pulled the rebar out of concrete. It would not have the stability and the strength, certainly not the tensile strength that it has with the rebar present. And this is what you see. You see these cubicles of mostly just lignin remaining. And in electron micrograph, this is what it would look like. It basically crumbles. So if you were to imagine which of these two would be, let's say, with a landscaping tree planted next to a house, I think it's pretty safe to assume that a brown rotter is going to present a much more imminent danger to a structure, a house, a car, or people if brown rot is taking place in a tree, since that tree is going to be a hazard much more quickly than, when I say quickly, I mean in terms of months or years, than a tree undergoing white rot. Some brown rotters include the oak maze polypore, this sulfur shelf, which is very commonly seen on stumps. This is a brown rotter. This seemingly innocuous, very crustose kind of basidiomycete. You're looking at the spore-bearing structure. So this would be the analogous, this portion of the fungus is analogous to the underside of polypore fungi, the spore-bearing structures. Velvet top fungus has the mellifluous name Phaeolus schweinitzii. And then I'm going to mention just for a moment here these other rotters, which are these blue stain fungi, these are pathogens. The fungi we looked at just a moment ago, they're rotters. And you could say that they are secondarily pathogens in the sense that even though they are, all of their metabolism is focused entirely on the dead part of the tree, which is the wood, to the extent that they compromise the strength of the tree, they can promote its demise through the tree coming down in a storm. So you could think of them as secondarily pathogens. These blue stain fungi, which will also rot wood, do come in and attack the vascular system as well. So these stain the wood blue, but they actually enter through the by parasitizing the vascular system. They're vectored by bark beetles, so they'll invade and infect phloem and xylem. So they are very, very different than the rotters we encountered just a moment ago. And they, therefore, as vascular disruptors, they're disrupting the water and nutrient transport. So these are aggressive pathogens. You might recognize these names, Ophiostoma and Ceratocystis. Ceratocystis was until recently the genus to which was assigned the oak wilt pathogen. Now it's still what it was. It's just been given a new, well, it's been assigned to a different genus, Bretziella. But Dutch elm disease is also in this group here. Then those three fungi I just mentioned a moment ago are not to be confused with the blue stain fungi, which are saprophytes. They just break down wood. You've probably come across pieces of wood on the ground and the forest floor stained this beautiful blue-green color, this teal color, and there's another example. These are two different very closely related species called Chlorosoboria. One is aeruginascens and the other one is aeruginosa. And its fruiting structures, when they are revealed, and they're not often found, are these in the group called discomycetes, showing the same bright teal color as the wood that they invade and stain. And these are not, again, not pathogens. They're only found on down wood. When I say down wood, I mean branches and that sort of thing. We call that coarse woody debris. All right, so trunk rots of conifers, another group of fungi, Fomitopsis, Pinicola, Phalinus, Ganoderma, we looked at a moment ago. These are important. These are almost entirely, but not exclusively, brown rotters. And they cause a lot of damage in the forest, especially in plantations, pine plantations, for example, down south, or pine plantations out west, where the fungus finds its way often through root grafts from one tree to another and compromises their strength so dramatically that these trees will come down, and as a result, their value for lumber is greatly diminished in the sense that they're worth absolutely nothing when they're grown for timber like this. Fomitopsis pinicola can colonize both gymnosperms and angiosperms. And this is another brown rotter that is found throughout the northern hemisphere. It forms these persistent conchs. Again, these are not the primary, these are outward manifestations of a much, much larger amount of biomass. In every case that you see these conchs and fungi growing off of trees, whether dead or alive. Phalinus pini this is a white rotter, so there's an exception there. This affects large spruce, pine, Doug fir, and causes a great deal of damage in young trees of virgin forest and also in plantations. Its decay is almost entirely, but not exclusively, confined to the heartwood. And so it has, the disease is called red ring rot. And it, again, dramatically reduces the value of timber. Ganoderma species, another white rotter. Ganoderma applanatum is the artist's conch fungus. I'll show you a picture of what I mean by that in a moment. This is a white rotter. It does cause tree failure over time. Some people use these for aids in climbing trees. Not a good idea because these conchs can break off. But it's called the artist conch fungus because the conchs are sometimes used by people. The white surface can be scraped away and used to reveal various people's artistic inclinations. Okay, so we talked about trunk and limb rots. I want to talk now about cankers and canker rots. I find these to be very interesting organisms. They're very important as pathogens in the forest. Cankers are any kind of localized necrosis of bark and cambium. So we're not talking about wood rotting any longer. We're talking about fungi that actively and aggressively invade the vascular system. So they are biotrophs, not necrotrophs. They cause necrosis of bark and cambium by living off of the living tissue of the tree, the bark and the cambium. The callus tissue formed as a result of these cankers is the tree's response to compartmentalizing the disease. Often, they're caused by the ability of fungi to enter through wounds, either wounds created by storm damage or human intervention or insect feeding. Any of these things can create avenues through which fungi can enter into the vascular system and initiate disease. And in many cases, this disease in trees can last for decades. So this is an example. These concentric rings of callus tissue, or the remains of callus activity, this is what we call a perennial canker, because this tree is continually dealing with this fungus as it perennially forms these rings to compartmentalize the disease. There's no rot taking place here. This is exclusively the result of a fungus invading the vascular tissue and eliciting in the tree the wound response known as compartmentalization through the formation of callus tissue. Some important canker diseases of northeastern forests, very famous ones, but we'll get to those in a minute. The annual fusarium cankers principally cause the disease of maples. Maples are an important crop tree, also important for producing maple syrup, of course. And then there are the perennial cankers, like I showed you a moment ago. Most famous of these, of course, is chestnut blight. That's formed by an ascomycete that causes canker. Eutypella canker of maples is easily seen or commonly seen in the forest. Target canker of birches, Neonectria canker, very commonly seen. And beech bark disease, which is another very famous disease that along with chestnut blight has had a major impact on the composition of the northeastern and eastern forests. Chestnut blight, this is what an early canker looks like. It eventually causes the bark to swell and erupt. Looked at closely, and you can actually, if you find chestnut in the forest, and it's not that hard to find young root sprouts, chestnuts that were killed decades ago are still sprouting root sprouts that don't make it very far, but you will find the cankers on these root sprouts, and you'll see this reddening, and that reddening is actually, these are all the tiny little fruit bodies of the ascomycete Cryphonectria parasitica that causes this disease. It kills these trees, or at least it kills the above ground portion of these trees. As I say, these trees can continue to root sprout for decades. Eutypella canker is restricted almost entirely to Acer species, maples, causes significant economic losses by causing these highly disfiguring cankers. It not only disrupts, because it disrupts water transport, it's going to compromise the ability of a tree to produce sap. And of course, it's going to compromise the tree's crown. And it does cause dieback of maples, including in young maples. This is an example of this sort of perennial canker formation that you see in these types of cankers. Another example. Target canker or perennial canker, or as it's continuing to be called by people in the forestry industry, Nectria canker. This fungus, Neonectria ditissima, and I'll talk in a moment about its sister species, Neonectria faginata, along with faginata, is native to North America. It has been in the genus Neonectria, not Nectria, for a long time, but the forestry industry refuses to stop calling them Nectria cankers. So that's where we're at with that. But anyway, this Neonectria ditissima has a broad host range of hardwoods. It is everywhere. It's native. It's currently most visible on birch. Didn't used to be the case, but as we lost chestnut, as we lost, well, we didn't lose beeches, but as we've altered the forest through eliminating natural control of deer, oaks are less abundant than they used to be in forests where birches are taking over. And with increasing density of birches, we see an increasing occurrence of these perennial cankers on black birch and yellow birch. But this fungus can also be found on beech, as it is a part of beech bark disease complex that I'll talk about in a minute. It is an important pathogen where it's not native in Europe and in New Zealand, of all places, where it causes a problem on the apple industry. So if apple, then therefore probably pear, and that is the case. And you can find Neonectria on maple, black walnut, and sassafras as well. So this is what Neonectria canker, the target cankers, look like. They can form multiple cankers up and down the stems, as you see in this young forest made up principally of red maple and a lot of black birch. Looked up close, the cankers can sometimes look like this. They don't often form this sort of classic target canker. Some people call these Muppet cankers because of their resemblance to Muppets. If you look really close to these cankers, you'll see these little red eruptions in the wood. Even closer look with a hand lens, you'll see these little red fruit bodies. These are analogous to a mushroom in the sense that just as a mushroom contains the spores that are the product of sexual reproduction, these little structures called perithecia bear the spores that are the products of sexual reproduction in this particular group of ascomycetes called Pyrenomycetes. So these are fruiting structures, but they have to be seen with a hand lens. They're bright red. I think they're beautiful. Beech bark disease is actually a disease complex, and this is a really important disease, as important as chestnut blight in the role that it's played in dramatically altering the composition of the eastern forest and the northeastern forest and the northern hardwood forest type. We have a little bit of that in Connecticut, down into the sort of northwest corner of the state. When I say the northern hardwood forest type, I mean the northern hardwood forest consisting principally of yellow birch, American beech, and sugar maple. This was initiated by the introduction of a scale insect from Europe on European beech, which is what you see planted principally as an ornamental tree in cemeteries, on town greens. Copper beech is a cultivar of European beech. That's Fagus sylvatica. And this potentiates invasion by a fungus that's native. So beeches and Neonectria ditissima and its sister species Neonectria faginata both co-evolved along with beech, but through the introduction of the scale insect, which creates little holes through which the spores of these Neonectria species can invade, have had a dramatic impact on eastern forests through this devastating effect on American beech. This map here shows the distribution of the native range of beech, which is the green. So beech, American beech can be found pretty far south, but in decreasing densities and numbers. It's most abundant in this area here that's pink, which is also showing the native occurrence or the natural occurrence of Neonectria species. You don't find Neonectria species much further south of here. The scale insect that's shown in this map right now is this blue area here. This is called the killing front of beech bark disease, the disease complex. So what happens is the scale insect comes in, creates all of these feeding holes, then the fungus moves in, which is where you'll see the Nectria now, along with some scale insect, but the scale insect first has to create the conditions for through which Nectria then enters and creates the cankers. So that's what that looks like, the range. This is an aftermath forest. So I mentioned the killing front, and then we have what's called the aftermath forest, where you principally have trees no longer really very conducive to scale insect feeding, just because there's not much room for scale here. But these trees, you know what beech looks like. Normally it's a smooth gray bark. These beech trees, this is from further north of here, like this would be found, can be found in New Hampshire, Maine, and then up into Canada, where these trees are heavily cankered. They're not dead, but they're barely living. And what happens is, the interesting phenomenon with beech bark disease is that beeches are a tree that are readily root sprout as a result of any sort of attack or compromise. If the parent tree starts to sense that it is in decline, it will sprout clones off of its roots. And so what happens in an aftermath forest is you actually lose tree species diversity because of these beech thickets that result, this abundance of these small canker trees. Many of these never reach fruiting stage. They just persist, and then they themselves will sprout roots. And now you have plenty of so there is still a population of a scale insect here, but for the most part, you just have the Nectrias coming in and creating an abundance of these cankers. So a little bit closer look here, and a much closer up look at these erumpent cankers, highly compromising the vasculature of the tree, and therefore its ability to mature. I'm just going to mention, as far as beeches are concerned, this is not a fungus, but it's new to Connecticut. This is a big part of my research right now, is beech leaf disease. This is caused by a foliar nematode. So a new direction for me. I am not a nematologist, but I'm doing my best to catch up. We as a group of forest pathologists have very little experience with foliar nematodes. Foliar nematodes are a group of pathogens that are known principally as a pathogen of vegetable crops and ornamental, herbaceous or perennial crops. But this is a whole new world for us, and something that we're still learning about, because it's a very new disease to North America, and certainly new to Connecticut. Anyway, back to fungi. So canker rots of hardwoods, these are wood decayers that attack sapwood, phloem, vascular cambium, entering wounds through branch stubs. Inonotus obliquus, this is canker rot of birch, is an example of a canker rotter. It is considered by some to be medicinal. This is not an endorsement, but when you hear people talk about chaga, this is what they're talking about. And again, just to emphasize, I am not endorsing this by putting this little thing up here. Okay, sapwood rot caused by Cerrena unicolor. This is, many people think this is turkey tail. It's not. It is another basidiomycete with a maize-like pore-bearing undersurface. This is a sapwood rotter, so this is a pathogen. Schizophyllum commune, which has its claim to fame, and if you studied genetics, if you had a class in genetics as an undergraduate, you probably learned some of the basics of genetics that was discovered as a result of the use of Schizophyllum commune being used to study very early on how genes are transmitted from generation to generation. A lot of this work having been done in medical research medical centers. And so for below ground, we talk about root and butt rotters, two principal ones, and one of these, Armillaria, has been somewhat in the news lately because of the major oak decline that we saw in the eastern part of Connecticut last year as a result of several years of gypsy moth defoliation, brought on or incited by several years of drought. And what happened was, as Armillaria is everywhere, these are native, they're soil-borne basidiomycetes, all the Armillaria species, and they were able to take advantage of trees that were heavily compromised, and where gypsy moths didn't kill the trees, the Armillaria and the two-line chestnut borers did. Tens of thousands of oaks in eastern Connecticut having succumbed to this sort of succession of onslaughts that took a lot of oaks down. Heterobasidion species, another group of root rotters. Armillaria root rot is often called the shoestring root rot fungus. There are at least 40 species, and they vary in aggressiveness and disease type and host preference or specificity. It's often also called the honey mushroom fungus. But where there's Armillaria, there's going to be pretty serious tree disease. These will infect both the cambium, and then will go on to then break down wood as well. So they are both a pathogen and a wood rotter. If you peel back bark, sometimes you can easily do it with your hands in advanced stages of decay, and you'll see this white weft of mycelium on the surface of the wood. Additionally, you'll see these hard string-like structures. Here's a better example of the white mycelium and the string-like structures. That's why it's called the shoestring root rot. These are called rhizomorphs, and they are a giveaway that you won't always see the mushrooms. You often won't. But if you see this and you see the shoestrings, you know that Armillaria is there and that Armillaria has invaded this tree. And again, these are pretty aggressive, can be pretty aggressive pathogens. Heterobasidion, also known as anosum root rot, named after one of the species of Heterobasidion, occurs in coniferous and mixed forests and has symptoms that vary by host. But you can see here the breakdown of this wood very aggressively taking place by this Heterobasidion. Again, a very important pathogen of forest plantations, or where forests are playing a role in producing lumber out west and in the south as well, and up in Maine as well. Now, fungi also cause a lot of foliar diseases, and I haven't even touched on this. These cause infection usually by entering stomates, not through woods in leaves. Sometimes the symptoms can be confused with insect feeding. What you're looking at here is a fungus that belongs to a group of very interesting fungi called rusts. And there are thousands of examples of this. They're very important. These are not rusts, but these are a various group of needle blights, a group of many different species of fungi. This is a huge topic and another topic for another time, because there's just too many examples to go into, but it's a fascinating area. Often vexes arborists working in landscapes because of all the blights that they encounter in Christmas tree plantations due to the rather unhealthy conditions in which Christmas trees are grown. You see a lot of examples of root rotters as well as foliar pathogens. So we've been talking about fungi as, I would consider the wood rotters to be friends of the forest, because they assist in the cycling of nutrients. When a tree falls, there may be no one to hear it, but the forest benefits from all of these processes. But there are other ways in which fungi are friends of the forest, and that is as insect pathogens. I mentioned gypsy moth defoliation, and also as mutualist symbionts. So mycorrhizae form these symbiotic relationships with the roots of plants as well as, and that includes trees. Most plant species are engaged in some sort of a mycorrhizal association, not all, but most. And wood rotters, as I said, I consider them to be friends of the forest because of the role they play in nutrient cycling and in moving trees that are compromised in one way or another. So they play a critically important role in forest ecology through recycling and restoring nutrients, and also through creating animal habitats. I mentioned the fungus that controls gypsy moth. This is the fungus Entomophaga maimaiga. This is in a group of fungal insect pathogens called the Entomopthorales group of fungi. This species was introduced from Asia, which is where gypsy moths came from, introduced in 1910 and again in 1911, and never to be seen again until 1989. It was assumed that it was a failed introduction, but lo and behold, in 1989, forests of the Northeast, people were seeing gypsy moth cadavers hanging from trees, and it was soon discovered that this was indeed Entomophaga maimaiga, having one way or another taking its sweet old time until 1989 to reappear and become an effective biocontrol of gypsy moths. Effective so long as we have a wet spring, that is. But this is what gypsy moth cadavers look like when they become infected with Entomophaga. Symbiotic mutualists between fungi and trees, the mycorrhizae. These are found in 95% of plant families and are broken down into two groups. There are the endomycorrhizae, so intra, endo means inside. These are the ancestral mycorrhizae, a relationship that was formed about 500 million years ago with plants, now found in 85% of plant families. And then the ectomycorrhizae, these evolved more recently. And these are extracellular, ecto meaning on the outside of, these are extracellular. And I'll show you a depiction of what's meant by this distinction. And these are found in only about 10% of plant families, but big plant families. And we'll see what I mean by that in a minute as well. These are so important that an issue in March 2015 of the research journal New Phytologist devoted an entire issue to mycorrhizae. So the ectos here are on the left of this. This is, imagine a root tip. So this is a root tip. These are the root hairs. These are root hairs, fine root hairs coming off of a root tip. And the ectos grow in between the cells of the plant. So they obtain their part of the exchange across cell walls of the plant and across cell walls of the fungi, and create this mantle around the root tip. The endomycorrhizae, formerly known as arbuscular mycorrhizae, through the formation of these arbuscules, they actually invade the root cells, the cells of the root, and they actually obtain their nutrients by living inside of the cells of the root. And so these are called, that's why they're called endomycorrhizae, formerly arbuscular. This is what ectomycorrhizal associations look like. You're not looking right now at the root. You're looking at a lot of fungus growing around the root. This would be half the diameter were it not for the buildup of fungal mycelium growing around these root tips. So they play a very, very important role in obtaining water and nutrients, and then obtaining sugars as an exchange. It's a barter system. And fungi such as this bolete, Boletus edulis, this is one of many mushroom-forming fungi, basidiomycetes, that exist in the forest as a result of a mycorrhizal association. The endomycorrhizae, despite their greater abundance, actually they do not form any kind of above ground gross morphology like these fungi, they're pretty non-distinct. There's nothing very charismatic really about these, but they are very important to many fungi, many plants, including, for example, orchids. And this is a microscopic picture of these arbuscules growing inside this root. And these are their equivalent spore-bearing structures, as I say, not nearly as charismatic as these ectomycorrhizal fungi, but certainly very important. So just a few more of these important mycorrhizal fungi. This is Russula emetica, not one that people who collect edible fungi would not eat unless they were interested in something that served as an emetic. Amanita muscaria, also, this is a highly poisonous but important mycorrhizal fungus. This is Boletus edulus. This is one that mushroom hunters definitely go after. And this beauty is a Violacea. I can't remember the genus right now off the top of my head, but it is, we don't see it very often here, but it is the color really is as dramatic as that. And then, of course, truffles. These are one of the few hypogeous or growing underground fungi of the many that exist underground that are considered edible. So fungi as friends of the forest, they create habitat for various animals, bats, bees, and owls, and of course raccoons. So they do play a role in creating these cavities, which can, exist. A tree can exist without all of its wood for a long time, and these cavities do play an important role. Unfortunately, I don't even want to show a picture of one of these trees that went up in flames in one of the more recent fires in California, sadly enough. And that's the end of my talk. I will take, let's say, 5 or 10 minutes of questions. If Dennis, you want to read any questions people have through the chat, I'd be happy to take them. You'll have to unmute yourself.
I think I did, didn't I?
Okay.
Thanks, Bob.
You're welcome.
So we have a couple questions on chat. Has evidence been found of fungi in some form in older ancient wood, like architectural digs, archaeological digs, or even petrified wood?
Probably not. Sometimes the staining, it's not going to be found in petrified wood, but it's possible that the remains of fungal invasion would be, there would be evidence, let's say a footprint in quotations of fungi, which would have allowed the wood to become petrified. The invasion of the various minerals and things that result in the petrification process may have been facilitated by some decay taking place. But again, I don't know that you would actually find fungi are very hard to find fossilized. They're almost non-existent. They just don't exist in that form. That's the best I can do for an answer. Sorry.
Okay. Can target cancer occur in dogwood?
No, I have not seen target canker on dogwood, and it's not believed to be a host. Dogwood's principle, we don't really see much in the way of root rotters of dogwood main disease of dogwood is a fungus, but it's dogwood anthracnose, Discula destructans, and that's a foliar disease, but can actually attack the twigs and buds, so you get a dieback with dogwood anthracnose. It's why we don't see as much. It was an introduced invasive, and it's why it's harder to see dogwood in our understories compared to what we used to see, and why so many people now plant. There are some somewhat tolerant or resistant varieties of native American flowering dogwood, but it's why you see so many people planting these Asian dogwoods like Kuzas, because they are tolerant of dogwood anthracnose.
Is there anything that one can do to protect, say, a chestnut from canker?
Not really. In Europe, where it just so happens that their introduction of the chestnut blight fungus, Nectria parasitica, involved a much smaller amount of genetic diversity, and they were able to employ a virus of the fungus that reduces its virulence. And the virus was able to spread on its own, but with increased genetic diversity, which we have in this country of the chestnut blight fungus, comes an increase in barriers to what's called anastomosis between hyphae. The hyphae have to be of the same genetic group in order to be able to exchange cellular contents, and that is the principal way in which the virus gets from one fungal isolate to another. So efforts that were carried out here in Connecticut by our now retired former chestnut blight pathogen person, Sandra Nagnestakis, she and others in other states tried very, very hard to employ or deploy the virus, but it was not successful because of the great deal of genetic diversity that prevents fungal anastomosis between strains. So the only solution at this point is the planting and reintroduction of either hybrids or genetically engineered chestnuts that are resistant or tolerant to chestnut blight. There is a researcher at SUNY ESF who has developed through genetic engineering, it's controversial, but they are resistant, and there has been some effort to breed backwards through Asian chestnuts and selectively favor the Asian resistance characteristic while maintaining as much of the American chestnut phenotype with varying degrees of success. It's a very painstaking process, but there has been some success. And reintroduction into the forest, though, is not likely going to take place at any kind of a scale that would satisfy anybody living today, because as chestnuts were the dominant canopy species of American forests, and they do not tolerate shade. So chestnuts will only reintroduce where they have a light break, a gap in the forest. And that's why you'll often see where there's a clear cut, where been a microblast or something that's taken trees down, you'll often see little chestnut sprouts coming up. And in the absence of chestnut blight, those chestnut sprouts would grow rapidly and become canopy dominants. And I say dominance meaning dominance. They are the dominant tree and would have been the canopy dominant of a mature forest in the past. But protecting no, once a tree is infected with chestnut blight, it's not, there's nothing you can do to save that tree.
If fungi are growing on a large beech near my house, is the tree a danger?
Can be, as any well-trained arborist will tell you, every tree is a hazard tree. And we're not facetious when we say that. If it's a beech tree growing near your house, there are things that could happen beyond the obvious that could cause that tree to lose a limb and cause damage. Could be a storm, could be straight line winds, could be rot that you have no evidence of that's taking out most of the wood. Remember that wood plays the principal structural role in keeping that tree erect. And that's certainly true with a lateral branch. The attachment of that lateral branch to the main stem is very, very dependent on a very strong union between the wood of the branch and the wood of the main stem. And if anything compromises that branch and all of the weight that it bears could come down. If you have fungi growing off of a beech tree, it does mean that there's some amount of internal decay. And the only way to assess the degree to which there is decay would be to have a certified arborist make an evaluation for you. But if there's no way you would have a fungus growing off of a tree and not have decay taking place, because that's what is resulting in that, in that fruiting structure. As I said, the fruiting structure is just a small amount of the biomass of the fungus that results in that fruiting structure, whether it's a conch or a mushroom or some other manifestation of the fungus. So the best thing to do is to have someone evaluate your tree to determine if it poses a larger than average hazard to the house.
And this looks like the last question in the chat. What will Connecticut, you have to put your futurist hat on for this.
Okay.
What will Connecticut's forest look like in, say, 20 years? What will be the mix of trees?
See, that's an easy question to answer because 20 years is just a blink of an eye for a forest. And so I don't anticipate seeing dramatic changes to our forest. I mean, but then again, it's hard to say. We don't know how beech leaf disease is going to impact the parts of our forest that have beech, which is a lot of our forests in the Northeast here and in Connecticut. If we were to have the same sort of droughty springs that resulted in the gypsy moth magnification and defoliation that was seen in Eastern Connecticut, given that the lower part of Connecticut has not so much the northern forest type, but more of the oak hickory forest type, we could see a dramatic loss of mature oaks in the forests of Western Connecticut and lower Connecticut. And as a result of that, with the loss of those, as well as loss of a lot of ash, which we were losing before emerald ash borer. So I just want to make that point that we were seeing a general ash decline, complements of climate change, long before emerald ash borer first appeared. That's because of this, the warming has not been a sudden change. We've seen an uptick in temperatures year after year after year, and ash are not tolerant of drought. Ash are not tolerant of heat. And so we've seen general ash decline, but we've lost ashes, we've lost elms, we've lost chestnut. These are changes that have taken place, I mean, except for chestnut blight, and I guess emerald ash borer. But I don't think in 20 years it's enough time to predict dramatic changes in our forest. I think we will continue to see red maples thrive. If you look at the range of red maple, it has the largest latitudinal range of any forest in the the northern hemisphere. There are red maples growing in Florida and Texas, all the way up into the northern parts of Canada. So we will not lose red maples. We will not lose birches. They're very tolerant species, even with their perennial cankers. God help us if ailanthus spreads any more than it has, because that could take over our forests as it has in Western Pennsylvania. So let's hope that doesn't happen. And you know, we might see a northward expansion of hickory and oak, but one has to remember that although we are moving into climates that are more typical of climates found south of us, the tilt of the planet relative to the sun does not change. So day length is not going to change. So we can't just imagine that trees and plants that are adapted to latitudes south of us could readily, necessarily at least, adapt to our climate because of the changes that we have in day length relative to areas south of us. So I think that's as much as I can say about changes in our forest in 20 years. 100 years, harder to say. And a lot of that is going to depend on us and what we as a species decide to do collectively and independently about our tendency to our habits of putting more and more carbon into the atmosphere. And I don't mean to be preachy, but that is really a deciding factor, another variable that we have some control over. So I'll leave it at that.
Can I give you one last quick question?
Yeah.
I think this will be fast. What if a fungus is growing about 3 feet from a tree? Does that pose a danger?
Depends on the fungus. It could be a mycorrhizal fungus, which in that case, it actually is good for the tree. It could have nothing to do with the tree, or it could be a root or butt rotter that's decided to form its, as we call it, fructification, its fruiting body, mushroom in common parlance, emanating from a root that's close to the soil surface. But it depends on the species. Obviously, a tree needs its roots, and if the roots are compromised, a tree can fail. And by fail, I mean come down. And that is not that uncommon. Any of those rotters, especially the brown rotters that I showed you, and especially Laetiparus sulphureus, the sulfur shelf fungus, will also invade roots as readily as it invades above ground wood. As far as these fungi are concerned, wood is wood. And so they will often move however they got into the tree, they will slowly over the course of decades move up and down and out wherever they can find wood that they can metabolize. So that's a question that would require, first of all, knowing what the fungus is, and making other evaluations of the tree.
Okay, that's it.
All right.
Thanks, Bob. Thanks a lot.
You're very, very welcome.
That was most enjoyable.
Good.
And, thanks everybody for joining us, and stay tuned for information about our next program.
Good night, everybody.